Oh my god! Bye bye!
Let's put my laksa, this is terrible!
No worries, Nicole. We can clean it up with a little water.
Hey Nicole, what are you doing?
Oh, washing off the stain from just now.
With water only?
Yeah?
Are you sure it will work?
Huh, but Daryl told me it will work. Like, he said I put in water, then the stain will come off already.
And you have been washing for 2 hours? Why don't you try a little detergent?
This looks like magic!
Here are the pictures of Nicole's shirt before and after washing. Let us try another experiment. Firstly, we add milk to the petri dish. Then we add food dye to the milk. Lastly, we add a tiny drop of detergent. Watch the magic!
So, how does detergent work? Let us start with an example of a soap molecule. Here, you see a molecule called sodium stearate. Sodium stearate is a common compound found in soap. Now, sodium stearate is an amphipathic molecule as it has a non-polar hydrophobic end, which is the long hydrocarbon chain, and a polar hydrophilic end, which is the carboxylate group shown here. Let us represent this molecule with a diagram. The yellow circle represents the hydrophilic head, and the lines represent the hydrophobic tail. The polar hydrophilic head has an affinity for water and is hence able to react with polar water molecules through hydrogen bonds, while the non-polar hydrophobic end is able to react with the non-polar oil or grease molecules by dispersion forces. Soaps and detergents are thus known as surfactants as they form the bridge between polar and non-polar molecules. When detergent comes into contact with grease, detergent molecules form a monolayer surrounding the grease molecule, and their hydrophobic tails react with the grease. The hydrophilic ends react with the water molecules in the aqueous environment, allowing the entire structure to be suspended in water. This results in the formation of a spherical ball-like structure known as a micelle. The micelle contains surfactant molecules, in our case soap and detergent molecules, which form a spherical structure to surround the non-polar molecules such as oil or grease molecules. Non-polar substances such as grease and oil are held inside the micelle and will be washed away by water. So what happened to the milk? Well, milk contains fat molecules that react with the detergent in the same way as grease does. This reaction causes the spinning and twisting of the soap and fat molecules, giving the effect shown in the video. The food dye was added to help make the movement of the milk molecules visible. So another application of this is our cell surface membrane. The molecules in our cell surface membrane form a phospholipid bilayer as shown in the diagram. So the phospholipid bilayer actually has two layers of lipid structures. The hydrophilic heads of the phospholipid bilayer face either the extracellular tissue fluid or the cytoplasm, and the hydrophobic tails form the non-polar interior core of the phospholipid bilayer. This structure of our cell surface membrane explains the selective nature of our cell surface membrane towards passive processes such as diffusion. For example, ions carry charges and thus cannot undergo simple diffusion across the phospholipid bilayer of the cell membrane due to the non-polar hydrophobic core. They have to undergo facilitated diffusion through a carrier protein. And this is the magic behind soap.